Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 139 for the podcast. This week, I'm in conversation with Christopher Duma of Regeneration Biomedical, a Newport Beach-based clinical stage company specializing in innovative stem cell therapies for neurodegenerative diseases. I hope you enjoy my interview with Regeneration Biomedical's president and founder, Dr. Christopher Duma. Christopher, welcome. Maybe you can start by telling us a little about your journey in medicine. You haven't always been running a biotech, that's for sure.
No, that's very, very true, Dylan. Thank you for the invitation. I think when I was 9 or 10 years old, I wanted to be a doctor. I wanted to help people. I had a family physician that was a great role model. And that led me down the path to definitely be pre-med when I was in college. I was fascinated by the brain, and I was a neurosciences major. So the interest in the brain started as early as, when I was in my late teens. Finishing college, went to medical school. That was a definite. I was thinking to be a neurologist. But as you move on through medical school, you realize whether you're a medical type or a surgical type, they have very different personalities, and I was the surgical type. So neurosurgery was a perfect solution for me. I went on to do my neurosurgical training at Georgetown University in Washington, D.C., and I then went and did a small stint in England, by the way, 6 months at the Atkinson Morley's Hospital in England. And this was a brilliant 6 months I spent with the famed Henry Marsh, who wrote a book called First Do No Harm. So I have been really blessed with having a very positive experience in the field of neurosurgery, and brought that to the West Coast in California, and have been practicing as an active neurosurgeon for the last 30 years.
So in those 30 years, there must have been some big evolutions in the field, some big changes. What are the ones that really stand out to you?
Oh, yeah. It really it has been fascinating. First of all, the technology that we use in the operating room is massively different than it was 30 years ago. Microscopes have become more intuitive. They have image injection into the oculars of the microscope, so you're actually seeing MRI images in your oculars of the microscope while you're operating on the brain. We have 3D guidance now, computer guidance with various systems like the Stealth system and surgical theater, 3D rendering of brains, and the 3D rendering models can be injected over the patient's brain while we're operating. I can actually see through a tumor and know that there's a large artery or a vein below it to watch out for. It's really advanced significantly over the years, and it's really more getting toward the concept of AI and 3D modeling in the operating room now.
Well, what were some of the projects or the breakthroughs that you worked on yourself in neurosurgery over these recent years?
Well, that's a very good question. I have my holy grail is to treat this horrible disease called glioblastoma, which is the most malignant brain tumor that exists in the brain. It is a incurable disease. Your survival is 14 months once you get the diagnosis. That's the median. So one of my missions was to figure something out, something different in the treatment of that. And that's another disease where we've had no traction in the treatment of that disease for about 40 years, 40. And it's been boggling the minds of neurologists, neurosurgeons, oncologists for that long. And we still don't have a control for that. I use a machine called the gamma knife, which is focused radiation, and I use it in a special way where we treat along the tracks where the tumor is going to migrate through the brain. So we basically treat it as if we're cutting it off at the pass, stopping the last migrating cell getting through the brain. And that has a United States patent, and it is a way that I treat glioblastoma to this day.
This is something a little bit different, though, than what you're working on with Regeneration Biomedical. What's the vision behind Regeneration? What's the vision behind this new company?
Yeah, well, it stems from the same need, my need to fix a problem, and the problem is that we haven't had a new treatment for the neurodegenerative diseases, Alzheimer's disease, ALS, traumatic brain injury, Parkinson's disease. We have not had a new treatment for those in about 20 years. Recently, in the last year or two, there have been two biopharma companies that have put out a couple of monoclonal antibodies that are at least targeting amyloid plaques in Alzheimer's disease. But unfortunately, they're not really disease modifying. They don't improve the patient's cognition. They just slow the deterioration. So, our company that I formed in 2018 is targeting these diseases that have an unmet need for new and different therapies. We're targeting all of these diseases using stem cells instead of a single targeting factor. For example, a multifaceted approach to treating these diseases. It's my philosophy and the company's philosophy that these diseases do not have a single targetable problem. Like at Alzheimer's disease, do you think there's one problem? If you remove the amyloid plaque, you still have some tangles to remove that are phosphorylated tau. There are other problems going on. There's problems with mitochondrial dysfunction, oxidative stress, membrane transport problems, inflammatory issues. Can a single pill or a single IV injection really target all of those at once? And so our philosophy is that maybe a stem cell can do that. That's kind of their mission. They can target multiple things at once. So why not use stem cells to target these dreaded diseases? And by the way we use stem cells and our actual product is used in a very novel way. We actually are the first in mankind to inject the stem cells directly into the brain for Alzheimer's disease, for example. So we're not giving an IV, we're not giving an oral drug. We're trying to defeat the blood-brain barrier by direct injection into the brain.
Was there a particular patient case, or was there a particular personal experience, something back in 2018 that gave you the impetus to launch into Regeneration Biomedical?
No, it wasn't a single. It was probably thousands. Over my years in as a neurosurgeon, thousands of people come across my desk in consultation that have Alzheimer's disease, that have ALS, that have MS, that have stroke, Parkinson's disease, and I just sit back and I say, there's got to be something for these people, and that was the light bulb that lit up over my head back in 2018, that we really have to do something about this and use the FDA to do it. I had done some research prior to 2018 in rats, an IRB approach in humans that actually had some merit to it, and we realized that we can inject things into the brain and let them do their job. So that was the light bulb that went over my head at that time. And obviously, if you're going FDA, one of the reasons to go FDA is to get legitimacy. There's a lot of illegitimacy in the field of stem cell work. get legitimacy and to be guided by the FDA. And I had to inject pigs en route to get through FDA clearance for this, and I I put little ports in pigs' heads, and then we injected our stem cell product, and the FDA liked that, and believe it or not, they gave us clearance for a phase 1 trial. So since 2021, 2022, we received FDA clearance to actually do this work in humans. So we're the first, really, that we know of that are doing this.
Maybe you can just or I can take a step back and just ask for a bit more of an overview of where you are when it comes to research and development, the pipeline there. You're talking about these injections and being able to work with humans there. Can you give me an overview of what the pipeline looks like, of what the research is right now?
Yeah, absolutely. So as I mentioned, when we actually proposed this to the FDA, that was in 2021. They gave us a laundry list of things to check off, and we did that. We satisfied that laundry list, and then that took about a year. And then they came back to us and said, "Well, we still want you to do work in large mammals." A Yucatan mini pig is a large animal, weighs about 70 pounds. And so I went down to a vivarium in San Diego and did that work. Those animals were sacrificed. They sent to a separate lab, and the results of the analysis went to the FDA, and the FDA said, "Okay, you're cleared for a human trial." So when you start your human trial, you need a sponsoring site. You need a hospital that would be willing to do the research. And my home hospital here, Hoag Memorial Hospital Presbyterian here in Newport Beach, was very open to welcoming this research because there hadn't been anything like it. So they gave the green light for the research to be done here. The FDA said you only need 9 patients to do the phase 1. And phase 1 is typically, and as is ours, a safety trial. So we're looking purely for safety and not as a primary endpoint. We are also looking for efficacy on the side. But we started our first injection of our very first patient in March of 2024, so last year. So our very first injected patient is out almost 1 year now. And it's been quite dramatic what we've seen and what we've experienced. Since that time, we've injected 4 others. So we have 5 total injected patients. We have 4 more patients in the pipeline ready to go because of the need for 9 patients. And of the 5 patients, not one has had even a headache after the injection of the cells into the brain. That's the pipeline. So that's where we are. We've finished our 5th patient, and we're moving to the 6th patient. And to continue the pipeline, we expect to be complete with the phase 1 by the middle of this year, and then we will propose to the FDA a phase 2 trial, which will be multi-institution, placebo-controlled, and move on with about 70 patients.
Right, so they're really the next milestones in the clinic there, and you're looking to do that really within the next year, next 2 years.
Absolutely, yeah. I mean, time is of the essence. I mean, it's like David and Goliath. We're David in the world of big pharma, and we'd like to move at their pace. It's incredible how quickly they get drugs out and do 1,000-patient studies in a matter of very few years. So we're in there with them.
And you're focused primarily or only on Alzheimer's now? There's scope to
Right.
expand that, right?
That's exactly right. We know that if we get approval from our phase 1 to move to a phase 2, that all of the data from the phase 1 can be used in phase 2 studies with all those other diseases. ALS, Parkinson's disease. We're thinking of working with the football players for a traumatic brain injury to give them some hope. So the only thing holding us back from that is money. It's very expensive to do these trials. To do a single patient is about $150,000 for the phase 1 trial. So you do the math, for 70 patients, it gets quite expensive. So we're seeking funding for this, and we're moving forth. But, we don't have the deep pockets of big pharma.
Can you walk me through a little bit how your particular stem cell therapy works? Because you're actually taking these stem cells from the individual, and is it from fat cells, if I understand correctly?
That is correct. So there are numerous sources of stem cells. You can get bone marrow, you can get stem cells from umbilical cord, you can get it from fetuses, you can get it from your own fat. And I think, first of all, no one is really averse to having a small liposuction, are they? And secondly, the fat cells are absolutely useful in this particular instance as well. So there's nothing special about the stem cells that come from other sources. But the most special thing about our stem cells is they're coming from the patient themselves. So we're not taking stem cells off the shelf. We're not taking what's called allogeneic stem cells and putting them into the brain, and nor would I even fantasize about doing that first before using the patient's own stem cells. So the stem cells that we acquire are from the patient themselves, a little liposuction. They get out about 50 cc's of cells, and those cells go to our laboratory, and then they are separated into the stem cell components. We use the vascular component of the stem cells, which is much more likely to affect neural lineages. And we prepare them in such a way that we choose the ones that are expressing something called Wnt, W-N-T. So I don't want to, spill the beans of what our secret sauce is, but our proprietary information here. But Wnt is a activating signaling protein that's seen on stem cells. And when Wnt is expressed by a stem cell, it actually acts as an on-off switch for other stem cells to turn on. And the stem cells will turn each other on by this expression of Wnt, and that's called the Wnt cascade. So if we can get the Wnt cascade going in the brain to target and help any diseased areas of brain, that's kind of the mechanism of action. And the second mechanism of action is that we are all aware, or not everybody, that there are stem cells that are living in our brains right now. So you, Dylan, and I, and everybody listening to you, we have stem cells in our brains right now. And believe it or not, they live right in the wall of the hollow cavities of the brain called ventricles. So if we were to inject our cells into the ventricles of the brain, we would expect and hope that our stem cells will percolate into those little stem cell areas that are already sitting there and turn on those latent stem cells that are just naturally there in all of our brains to get to wake up and get moving. So it's kind of a paracrine effect that we're expecting. We don't expect a direct effect from our stem cells, but the beauty is that the brain is sitting there, even the brain of an 80-year-old is sitting there with stem cells waiting to be turned on.
You mentioned before you're bypassing the blood-brain barrier by injecting directly into the brain. Why is it really important to bypass that barrier when you're treating a neurodegenerative disease?
Yeah, So that's been again, the holy grail for the field of pharmaceuticals, getting through that dreaded blood-brain barrier. And it's Mother Nature's way of protecting our brains. It's a beautiful mechanism that it really saves us from all sorts of tyrannous problems that can happen in our lives, in our daily lives. If you ingest something, it's not going to get through that blood-brain barrier. If you take a poison, it might not get into that blood-brain barrier. Even if you have something administered IV, it may not get through that blood-brain barrier. But there is another barrier, and it's called the CSF ependymal barrier, and that barrier is literally the barrier between the hollow cavities of our brain, called ventricles. A lot of people don't realize that our brains are hollow. It's like a honeydew melon. You cut the melon in half, and the center is hollow. If you put a tube in there and inject stem cells, the layer between the brain itself and the ventricle, the hollow cavity, actually becomes porous when we get older. So when we're young and thriving youngsters and babies, that barrier there, the wall of the ependyma, has very tight junctions. Interestingly, as we age, those junctions get less and less tight, and they become actually porous enough for a stem cell to eke through. And so we know from our rat work that the cells injected into the ventricle can actually percolate through these little porous defects in the ependyma and wend their way into the brain. And they're being delivered there by cerebrospinal fluid. The fluid is floating through every area, every nook and cranny of the brain. So if you want to deliver stem cells everywhere, including the spinal cord, it's a great way of doing it. And it's our invention. It's actually, frankly, my invention. And we have also a U.S. patent for that. So that is our novel way of delivering the stem cells into the brain. Exactly what happens for an Alzheimer's patient after that, we don't know. We're going to figure that out, though, with CSF analyses going forward on all of our patients.
Recruiting these patients for a phase 2 trial, are there going to be any ideal candidates there, or any limitations placed on participants in a trial like that?
Yeah. So in our phase 1, we're accepting patients between the ages of 45 and 80. So that's a very large range. But we're only accepting patients who have mild to moderate Alzheimer's disease. The big pharma studies that just finished used patients that were mild cognitive impairment. That's a stage 3 patient, and stage 4 patients. We're using stage 4 and 5 patients in the Alzheimer's group. Alzheimer's has 7 stages of the disease, and obviously stage 7 is morbid. But the difficulty in enrolling is there is no difficulty. The biggest difficulty is competing with other new drugs like donanemab and lecanemab that are out there right now. And in our study, we're not allowing anybody who's had those drugs to be in our study because, frankly, they cause edema and hemorrhage in the brain and brain shrinkage and have all sorts of side effects, and we don't want our data kind of affected by that.
And what about the treatment process? You talked about a period of time between the liposuction and the injection of the stem cells, but is that the extent of the treatment? Is there a secondary injection or any particular follow-up after that surgically?
Right. The plan is to inject the patients every 2 months with their cells, and that would be in a phase 2. Phase 1 that we're doing now is a single injection. And when we start discussing some of our secondary endpoints, that's going to become very exciting because with only a single injection, we're getting some amazing secondary effects that are are very promising. So the patient's process is exactly this. They first go through a liposuction, and that is about a 20-minute procedure, and it's a same-day procedure. They go home that day. Uh, it then, about 2 weeks later, they get a port implanted into their brain. It's called an Ommaya reservoir. And we've actually used Ommaya reservoirs for 40 years in the field of neurosurgery to deliver chemotherapy for brain cancer. If one is familiar with what's called a portacath, which is the port that's placed under your collarbone to deliver chemotherapy and is used over and over and over, we have the same thing for the brain. It's called an Ommaya reservoir, and that gets implanted. That is a 15-minute operation with extremely low risk. And once that's implanted, it's under the scalp, so no one sees it. And once that's in there, the tube that goes into the brain can deliver our stem cells over and over again. The fat that was taken on the first stage is worked on at our lab and then cryopreserved. So we sift out the Wnt expressing cells, we save them, store them, and a single liposuction should be good for an entire year of injection in a patient in a phase 2 trial every 2 months. So then,
go ahead.
Well, yeah, I mean, and then finally, once the cells are ready to be injected into the brain, which is about 4 weeks after, we work with them, then the cells are requested by the physician, and they come in a small syringe, and they are injected at the patient's bedside through the port in a 5-minute procedure. That doesn't hurt at all. Putting the needle into the port doesn't hurt, and the cells are injected, and then the patient goes home.
Let's take a step back and maybe look at a bigger picture here in the US. How would you describe the state of stem cell therapy research right now in the US? Is it advancing? Is it stuck? Is the new administration scaring people into not moving forward?
Well, I think that stem cell research has really gotten a bad name in the past. I mean, there's these rogue clinics that are using stem cells to treat the common cold has been a problem in the field of stem cell research. The only way, in my opinion, to legitimize that research is to get in with the FDA and use FDA cleared trials. But as far as changing the mentality of a patient, traveling to China or Germany to get a single stem cell procedure for God knows what at a exorbitant price, that still is going on. But as far as legitimate research that's going on like ours, we're very, very few are being done right now. There are a few that I know of that are actually injecting into the brain, but not like we are through the ventricles, and getting some promising results. But again, with a disease like Alzheimer's disease, MS, these are whole brain and spinal cord diseases. You need to get your stem cells everywhere in the brain, and you can't just target certain areas. So I think, to answer your question, we're getting there, and it's starting, and it's the beginning of what's called personalized medicine. Personalized medicine is probably the next iteration of the pharmaceutical industry. It's it's very easy to take a pill, but it may not be the solution for certain problems. I mean, you may have to get a hole drilled in your head and a port placed to treat Alzheimer's disease or any of the others.
You mentioned the cost for a phase 2 trial, a phase 2 clinical trial. Is the financial landscape or maybe the scientific landscape or the regulatory landscape, are these challenges for people in your position to overcome?
Well, you mentioned a few things there, the financial and regulatory, right? In terms of financing our work, our phase one trial has been financed by friends and family money. Almost everybody's been touched by Alzheimer's disease, and our first phase one trial is for Alzheimer's disease only. Uh, Everybody's had 6 degrees of separation of someone with Alzheimer's disease, family members, et cetera, and people are willing to donate or invest money into these projects. When we get into the phase two level, we're talking about a need of about $25 million to do a 70-patient phase two trial over a year and a half. So those numbers get a little bit dramatic, and still there are private investors who can come up with that. There's institutional money that can come up with that. There's investors and financiers who will do that, and what they do is they get stock in our company, and they see how we do as we move on as an investment. But right now, we have been seeking the Series B round of $25 million to be satisfied that we can fully start and not have to quit halfway through a phase two trial. So that's number one. And number two is the regulatory. I think I'm just fascinated that the FDA allowed us to go forth with this. And the real reason is, I think, is that there's an unmet need. We need to treat these diseases. A lot of them have absolutely no effective treatments. Number two, it's been decades since there's been any good treatments for these. And number three, if the stem cell world is now going to become something real, the FDA wants to regulate it, and I say that's okay. We want to keep the the legitimate people in the pipeline and the illegitimate ones out.
How does the US compare to other countries when it comes to stem cell therapies and the research and development there?
Yeah, I mean, there are a few other countries that are a little less, a little more lenient, less strict with stem cell work. Japan is one of them, and China is another. They're a little more open to stem cell research, but really that's about it. I mean, the European, I don't know much about the European, the European Medical Association, whether they're allowing stem cell work there, but I'm a little out of touch with that. But I think by virtue of our doing our work right now here in the US, I think they're looking to us. The FDA is looking to us to allow further studies to be done. And they're going to be very happy with the results we have.
In the past, stem cell therapies were sometimes considered a little controversial, depending on where the stem cells were brought in from. You don't have any of those problems when they come from a person's own fat cells.
Precisely. I mean, it's really a social issue, a personal issue when it comes to getting stem cells from aborted fetuses, for example, or from fetal tissue. So yes, we're not using that, and I didn't think that this FDA would even approve it. They wouldn't even come close to approving that as one of the first trials of its kind.
Let's take a look a little bit into the future now. Obviously, you're looking forward to phase 2 clinical trials, but if you were looking at a little bit further ahead, sort of the 5 or 10 year horizon, where do you see Regeneration Biomedical then?
Well, we have this vision of becoming the fathers of stem cell therapies for neurodegenerative diseases in the brain. We believe that we can move forward with the phase 2. Let me just tell you a little about the results of phase 1 that are not the primary endpoints. The secondary endpoints, out of the first 5 patients, we've seen actual cognitive improvement in their cognition. In 4 out of the 5, their ADAS-Cog scores went up, their mini mental status scores went up, their ADL scores went up. So that has not even been seen by big pharma with their 2 new drugs. We've also seen a decrease in the amyloid plaque density. We look at that using amyloid PET scans. So every patient in our study is getting a PET scan, and you can see the reduction of the amyloid plaques by half in our single dose for the patients. And by the way, we're also seeing the same results for phosphorylated tau, which is the tangles that are seen in Alzheimer's disease. Those have all normalized 100% in all 5 patients so far. And family members are saying, wow, our family member is doing great. They're much better. And by the way, in 1 year, like our first patient is out now 1 year, we should see some significant decline in their cognitive function, but they seem to be stable or improving. So that's really very promising. So we expect phase 2 to look the same. And then after phase 2, you make the decision whether to move to a phase 3 if you have enough money to do that, and investor money, et cetera, or big pharma may pick it up. Now, they may also pick it up now as a synergistic approach to a disease where they're, for example, using their monoclonal antibodies to clean up the debris of beta amyloid, that's the plaques, and then hitting it with a chaser of stem cells, such as ours, to build, repair really a lot of different mechanisms. The membrane transport is one of the biggest problems in these diseases, and protein folding is a big problem in these diseases, and perhaps the stem cells are good at prophylaxing against that. That's the short answer is we will see after phase 2 where we are.
Are you actively looking for big pharma partnerships, or are you still sticking with private fundraising?
No, we'd be delighted to entertain big pharma partnerships. Absolutely, I would never turn anything away at this point in our development. But I'm quite sure they're all probably waiting to see what phase 2 looks like.
You've been a researcher, you've been a surgeon, you're now on the business side as well, really an entrepreneur. What sort of advice could you give to someone who's really interested in neurodegenerative diseases and finding solutions? Where can they start? What should they be reading?
Yeah, well, I think interesting you should say what should they be reading. A lot of my work, a lot of my reading is done by signing up with Google Scholar. Google Scholar is a way where you can put in keywords, and Google Scholar will send you every week the latest scientific articles that have come out with those keywords in it. So, in my case, it's, stem cells, neurodegenerative diseases, and subventricular zone. That's the zone that has the stem cells in our brains innately. So I want to see where all the research is going. So that's what you read. That's what you look at, because it's literally the fastest information you can get. Obviously, listening to your podcast, Dylan, Beyond Biotech, is a source, another source for anybody very interested in this, and updated. I mean, you're talking to me live in a very updated fashion. You're hearing the latest even before we publish. The second is to go to scientific meetings. I have been submitting to various meetings locally here in the United States and internationally. We just got back from presenting in Madrid at CTAD, which is Clinical Trials for Alzheimer's Disease, and we got picked up by a podcast there, which is, by a digital news source there called Biospace. and they compared us with side by side with Biogen, Eli Lilly, and good old Regeneration Biomedical. So we got some real traction there. But interestingly, we just submitted to the ISCT, that's the International Society for Cell and Gene Therapy. The meeting is in New Orleans in May, and we are very proud to say that our abstract received an award. They only give about 20 awards out of about 1,500 abstracts, and we were one of them for our abstract submission. And we will be giving a podium presentation at that meeting in May. So that's a feather in our cap. And finally, I would say to any new researcher, think outside the box. I did, and believe it or not, by perseverance, the FDA approved it. So if you're thinking outside the box, you got to think of something new, novel, different. Go for it. And I would suggest that don't be shy, don't be bashful, and try something new.
Christopher Duma, thanks for joining us on Beyond Biotech.
Thanks very much for having me on.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.